Okay, well, welcome back, people, to a new year and to our first seminar for the 2025 season. Appreciate people who could make it given health issues and scheduling at this time of year, so appreciate it. As always, I need to remind you that these meetings are live streamed and recorded for future viewing, and so please be aware of that. As always, Tom, myself, Jim Sluka, and Lorenzo are available to take your suggestions and criticisms and ideas. So feel free to reach out to us at any time uh, if there are any issues that you want to address. We have various communication channels. They work better if they're used. We have a LinkedIn page. We have the YouTube channel. We have the IMAG MSM Wiki page. And we have also a Twitter handle if you want to use it, MSM Viral. If you want to share a publication in this area, we can put those up on the IMAG page, and we can also put those up on the GLIMPRINT website. And so there's also the GLIMPRINT website that's being worked on. So I'm not sure what the status of that is at the moment. I think there were some changes in the University of Nebraska management of the website over the Christmas holiday. So if you have any trouble with that, please reach out to Jim Sluka at iu.edu for help. Reinhard Labenbacher at University of Florida continues to search for faculty for his computational biomedicine initiative. Tom Helikar has multiple openings in computational immunology and is eager to hire. So if you're looking for a position, a postdoctoral level senior scientist, please reach out to Tom at the University of Nebraska. And Jason Shoemaker continues to recruit in influenza modeling. And so you can reach out to him if you are looking for a position as well. We've talked about these grants a lot. ARPA-H Catalyst is still looking for solicitations, for proposals. There are these new announcements from NSF, NIH, and FDA. There were some recent announcements specifically on NAMs from NICEDM in the United States for new opportunities in NAMs. And so those are potentially of interest to this community. The open source ecosystems has basically done for this year, January 14th, but that is supposed to continue during future years, and so maybe still of interest to people. There's also the NOSI on infectious disease modeling, and that will have the regular due dates for NIH going forward. The next one would be in February and then in the summer. There's still that workshop on virus dynamics in Bordeaux that people might be interested in a year from now. We will not have a steering committee meeting this week. Our first meeting will be on January 24th. And are there any other announcements to make before we continue here? Okay. Going forward, we have Adam Knapp and Chase coming up. We're looking for speakers for January 23rd and February 20th. You can suggest yourself, your students, people whose talks you've enjoyed hearing. Don't feel obliged to, even if you've spoken before, you can speak again. Lorenzo, do you want to say anything about the scheduling before we go forward?
No, nothing beside the usual invitation to anybody who feels like proposing themselves or their own students to present. They are welcome to write me or any of us.
If you've read a paper that you thought was interesting, please suggest the people. It's particularly interesting to us when people come in who are not the usual suspects, and very often that brings new people to the community and new opportunities. So please do help us out. Okay, so we now will move on to our talk. Uh, we have Martina Iulini from the Università degli Studi di Milano, who's going to talk to us about using NAMs to address PFAS immunotoxicity, very timely in the sense that it bridges toxicology and immune system, which is an area of great interest to this community. There are major initiatives in funding and in science for novel alternative methodologies, both in the United States and in Europe. And so this is an area really of importance, I think, to understand, and especially for those of us in the United States, to understand what's happening in Europe is really critical. So I will turn over the screen share to you, and we will look forward to hearing about this exciting work.
Okay, great. So I can start. So thank you so much for inviting me to join that seminar. I just present myself. I'm Martina Iulini from okay, no worries. I'm Martina Iulini from the University of Milano, and I am a PhD student. Actually, I'm just waiting for the defense, so I am in the last year, and I work in the Laboratory of Toxicology and Risk Assessment of UNIME under the supervision of Professor Emanuela Corsini. And today I talk about basically the results that we obtain after 2-year studies for a project granted by EFSA that have the aim to study the PFAS immunotoxicity using only new approach methodology. I try to go on. Okay, yes. So if you want to go more in detail about all the results that I present now to you, you can find the entire publication and report in the EFSA External Scientific Supporting Publication Journal, and here you have also the DOI. And as you can see here, is not only the work of our laboratory, but was also in collaboration with the University of Catania, also in Italy, and with the Wageningen Food Safety Research in the Netherlands, and with the SQ Lab, that is an industry that work with the in silico field. So first of all, just introduce, I think quite all of you know how PFAS are, but just to introduce a little bit. So PFAS are poly and perfluoroalkyl substances that are widely used in industry due to their surfactant properties. That properties are given by the carbonylate change that represent the base of all of that topology of chemical. Right now, EPA classify more than 40,000 different chemicals under the name of PFAS, and recently say that more than 600 are right now in the use in industry. So due to the presence of carbonylate bound, they have a higher stability in the environment, and since they are very used in industry, there is now environmental alarm since they are that stability is also present in the environment and it's not possible to degrade easily that chemicals. So since there is the environment release, they can accumulate with the food chain and also arrive to human, in which they are also classified as endocrine disruptor, and so they can affect only the immune system but also other organs. In 2020, in particular in Europe, but I know also in the US with the EPA, the EFSA in particular, so the Agency for the Food Safety Authority in Europe, established the tolerable weekly daily intake for PFOA, PFOS, and other true long-chain PFAS. Identify that and classify that threshold based on the impact on the immune system. And in particular, the major endpoint was the results, the effect on the efficacy of the vaccination. But Okay, the critical endpoint is not only the vaccination, but in literature you can find a lot of results also related to a general immunosuppression function and also linked with an increased risk of autoimmune disease, but also, for example, PFAS and was classified recently by the IARC as a carcinogenic compound and also PFAS as a possible carcinogenic compound. There are also some less evidence that connect the PFAS with inappropriate immunostimulation and increase of allergic phenomena. And as I told you before, stronger evidence are found on immunosuppressive effect. And all of them have a connection and are represented more by a reduction of the antibody release. So starting from that evidence that when we started in 2020, was based simple on animal study and some epidemiological human study, we decide to focus the attention of the antibody release and understand and study how the antibody can release in our immune system. So just briefly, the antibody can be released in two different ways. So we can have a simple T cells independent B cells activation in which only B cells are the actor of our response that recognize the antigen, mature to plasma cell and secrete antibody. While, in the other hand, it's possible also to have the interaction of more cells with the T cells dependent B cells activation, in which not only the B cells are the actor of our response, but we have also dendritic cells and T cells. So starting from that, we decide to study all the effect on all the single cells involved in the antibody release for both of the way, just to understand better which are the target of the effect on PFAS on the immune system. So the aim of the project granted by EFSA was the evaluation of the immunotoxic effect of PFAS and important also the identification of the mechanism behind that effect, using only alternative method to stay in line with the new technology toxicology view. So to do that, we try to design an integrating testing strategy based on in vitro method and in silico method. We use the in vitro method to try to mimic the in vivo evidence that was present in literature and also to evaluate the parameters that are relevant for the in vivo immunotoxicity. And we add the in silico method to cover the gap for which that we have in the in vitro system, and in particular to simulate the blood concentration exposure of PFAS in vivo. So starting from the in vitro strategy, we design a strategy, as I told you before, that analyze the effect of PFAS on the single cells involved on the antibody release. So before an assessment of the leukotoxicity to be sure to work with the safety concentration, so that not affect the cell viability, we decide to study the effect on dendritic cells, maturation, on T cells, differentiation and proliferation, on T cells, independent antibody production, try to introduce a new in vitro primary antibody response protocol, and also on T cells independent antibody production. Before to go in detail about that protocol that we decide to apply and also the results, here just to show you which chemical we decide to test, so which PFAS. So we decide to test the first more major PFAS, so PFOA, PFOS, PFNA, and PFHXS. First of all, because are the one for which we are more exposed right now, and also because are exactly the four for which EFSA based the threshold in Europe. And we decide to apply only in vitro human, only human cells, so T cells, THP1 cells that are monocyte, is a stabilized cell line, but also human peripheral blood mononuclear cells taken from healthy donor of both of the gender to increase the relevance for human and also to see if there is eventually a gender difference. Starting from the first cells that we decide to use, we try to see the effect on dendritic cells maturation. To do that, we decide to apply a protocol developed in 2005 by Berge et al. That used THP1 cells as a surrogate of mature dendritic cells. That one is a protocol that is use a lot and is a reproducible and robust method, and it was based starting from THP1, so from monocyte differentiated for 4 days with 5 days, sorry, with a cocktail of differentiation to obtain immature dendritic cells. After, to be sure to obtain immature dendritic cells, that we check it with flow cytometer analysis, we go on adding and expose our cells to the 4 different PFAS at increasing concentration that was selected based on not cytotoxic effect for 24 hours. After 24 hours of exposure with only PFAS, we add at our cells also a maturation cocktail to obtain a mature dendritic cells, and we analyze the effect after 24 and 72 hours of maturation. We decide to analyze the most important protein involved and present during the activation and maturation of dendritic cells, so the costimulatory pathway of CD86 and CD83, and also HLA-DR, that is part of the major histocompatibility complex, and then link the T cells, and also some cytokine. Related to the expression of the most important marker of the dendritic cells activation, here you can see the results. So you can see different color, each color represents the different PFAS, and also the statistics results match with the same color. As you can see, there is a different trend related to the effect of between the different PFAS, and also a different impact. So, for example, if we look in particular HLA-DR after 72 hours of expression, you can see that quite all of the PFAS was able to suppress the expression of that important marker after 72 hours. And in particular, that suppression it's possible to see also at 24 hours only for PFOA. So normally HLA-DR is active from a strong sensitizer at 24 hours and weak sensitizer at 72 hours. So we can assume that in that parameter for that endpoint, maybe PFOA was able to have a more potency related to that marker compared to the other one. And so we can see the reduction, so the suppression of that marker also at 24 hours instead of 72, that is the normal timing of expression of that marker, and so of reduction. Concerning the other parameter, you didn't see so much difference except for CD83, that is an important marker that is present in dendritic cells, mature and not immature. And so you can see that PFOS at all concentration tested and PFOA at the higher concentration was able to suppress that marker compared to the positive, compared to the negative control, so the mature dendritic cells. I just forgot to say, but I want to add now that the concentration that we use, so starting from nanogram milliliter to 10 microgram milliliter, are a very high range, but represent right now the range for which all of the people are exposed. So we decide to use that concentration also to mimic the in vivo concentration. Then, if we move to the second cells that are involved in the T cells dependent antibody response, we found the T cells. To study the effect on T cells, we decide to use the peripheral blood mononuclear cells of healthy donor of both of the gender, and we expose that cells to the different PFAS at increasing concentration for 24 hours, as we do also for the dendritic cells. And after 24 hours, we add a maturation cocktail of anti-CD3 and anti-CD28 to reach a total amount and activation of T cells for 4 days. And after that, we staining our cells to study the surface antigen and also intracellular one, and also collect some cytokine. But before to do that, we also analyze the effect on T cells proliferation. And here you can appreciate that increase the higher concentration of PFAS was able to suppress the proliferation of T cells. So if you look on the top, I can put, okay. If you look here, you can see that the G0 is the first population, while the G9 is the last population, so the new one. And you can see that increasing the concentration of PFAS, you can appreciate that the G0 peak increase, so more cells stay in the G0 compared to the new one, meaning that the higher concentration of PFAS was able to block the proliferation of T cells. And below you can see the results obtained for both of the gender, in which in that case you can see that the male seems to be a little bit more affected compared to, female compared to male one. Then, if we move on the T cells differentiation effect, so if we look in detail about the different subpopulation of T cells, you can see that we found and discovered at the end a general suppression of all the single T cell subpopulation. So we see a reduction on T helper 1 and connecting interferon gamma activation. The same we found also in T helper 2 and connective cytokine interleukin 4, the same for T helper 17 and interleukin 17, and also a downregulation of the Treg and its cytokine. That one we found for both of the gender. Here is represented the statistical results with the concentration that at the end was statistic, and you can see that in some case also the lower concentration was able to suppress the differentiation to the different subpopulation. And if you look at the ratio between T helper 1 and T helper 2, you can see that is a little bit more T helper 2 predominance, meaning that the effect in our condition is more related to humoral and allergic immunity. But we found at the end a general immunosuppression also for the T cells. Then we move to the last cells involved in the T cells dependent antibody response, that are the B cells. The gold standard right now for the identification of the chemical in a T cells dependent antibody response way, so the TDAR, are based on animal model. There are some models that use the PBNC, but we have the red blood chip. So in any case, there is animal. So starting from literature, a very old method from Komatsu et al., we try to develop a new protocol that don't use part of animal in the protocol to try to perform a in vitro primary antibody response without the use of part of animal. So to do that, we start again from our PBNC. We treated for 24 hours with increasing concentration of PFAS, as already we do for the other protocol, and then we stimulate with the KO lymphetemochanin plus the Staphylococcus aureus CoA1 that was able to induce the proliferation of B cells without the secretion of the antibody. After 5 days, we wash out our cells, and we had again the PFAS and also interleukin 2 for other 5 days. And at the end of the total 11 days, we analyzed the anti-KLH IgM to see how our PFAS affect that parameter. So as you can see here, the induction that we obtain compared to the unstimulated one is not so high. So for sure the next, and actually what we do now, is to better develop also that protocol, but you can see in any case that for all of the PFAS, for both male and female donor, you can see a decrease of the secretion of anti-KLH IgM compared to the control for quite all of the PFAS, and we reach in some cases a statistical significant reduction. That protocol have the problem that in our sample we didn't have a lot naive and T cells, so we need to find a better balance of cocktail to induce a higher proliferation, and then maybe appreciate more eventually reduction. But we are in any case very happy to try to develop also that protocol and without the use of animal. And the last one is the analyze the effect on T cells independent antibody production. So a little bit more easy because we use a method that was already standardized and developed in 2020 and was based again on TBNC treated for 24 hours with PFAS, and then we added the deoxynucleotide 2006 and interleukin 2. So in that case, since it's a toll-like receptor 9 agonist, we are able to induce the proliferation to plasma cells and also increase the release of the antibody. And in particular, we detected IgG and IgM. Here you can appreciate that in that case, the reduction compared to the control is high compared to the cells-dependent antibody response, and in particular, it's possible to see that seems to be more affected the female compared to the male. And in female donor, you can appreciate a reduction compared to the control already at the lower concentration, so 1 nanogram per ml for P4. Then, if you go more in detail about the single IgG and IgM release, again, you can appreciate that the female seems to be more affected compared male, but you can see also that in the female, the IgM seems to be more affected. And that one is in line with the literature results in which they see that the IgM of female donors is more effective of PFAS compared to the male one. So just a brief summary of the in vitro results. So we can say that starting from epidemiological data, we know that there is a general reduction of immune function, and in particular, the reduction have a focus on the antibody release. And if we go more in detail about our results, you can see that for both of the way we obtain a suppression of the immune system, we obtain immunosuppression that is more related, more mediated to HLA-DR for dendritic cells, general T cells immunosuppression with a more TLP2 predominance, and that suppression is both in the intracellular and extracellular cytokine, and we reach the reduction also in the IgM anti-KLH. And in the same way, we have a general reduction also on the antibody IgG and IgM. So we can conclude that the protocol that we decide apply, so the NAM that we selected, offered the opportunity to study the PFAS immunotoxicity and try to mimic and reproduce the epidemiological evidence. And starting from that, we think about, okay, but what are the limits of our in vitro method? So in the in vitro method, we are not able to take in consideration the kinetics of our substances. So to cover that gap, we decide to also add the in silico method. And to cover the kinetic, we decide to use the PBK model. So the PBK model is a mathematical model that is able to predict the ADME of the chemical of the substance, in our case in a human. And we was lucky because the EFSA in 2020 developed a code for study PFAS, in particular P4 and PFOS. And so starting from that code that was based on Lochisano et al. And was validated and evaluated by EFSA, we was able to also cover that gap. So we assume that our PFAS in our model was take from gut, so we assume oral exposure. And in the model, you can mimic from 0 to 1 here with a breast exposure and then with a dietary exposure. So you are very open to see all the different ages of and the impact. So we decided to run the model from 0 to 75 years old. And our patient receive the PFAS through food intake every 8 hours for the entire lives, and from 0 to 1 with the milk, and then with the dietary exposure. Here you can see the results. So you can see 3 different colors. Each color represents a different scenario. The black one represents the scenario that mimic the French study used in the EFSA Contemporaneous 2020 scientific opinion. So basically, for us, is the simulation of the oral concentration exposure for general people. Then the red one represent the EFSA threshold for the main for PFAS that was made on 2020 based on epidemiological data on the vaccination, and that threshold basically is right now applied in Europe. While the blue one represent the dietary exposure of people that live in contaminated area, and also that data is taken from epidemiological data present in the EFSA content panel. And you can see that we have an increase absorption from 0 to 1, represented from the bractic exposure, then a sort of slight decrease with an increase that is conservative all of the time for both of the red and black line, so for the ones that are in a safety zone, while for the blue one we have a continuing increase. So all in the scenario that represent the people that live in contaminated area, we have a continuing increase. And we measure the increase in plasma concentration level, and is in nanogram per ml, so you can see that we are in line also with the concentration that we use in the in vitro method. Sorry, so we start from 1 to nanogram per ml, and we reach the PFHXS with 80 nanogram per ml for the occupational exposure, the people that live in occupational contaminated area. Then we decide to use the data obtained from the PBK model to inform the Universal Immune System Simulator. The Universal Immune System Simulator in collaboration with the University of Catania. And if you want to go a little bit more into also that, you can find all of the results not only in the EFSA report but also in that publication. And we do that to increase and add also the dynamic of the modeling and increase the relevance for human. What is the Universal Immune Simulator? So is a mechanistic computational platform that is able to simulate the immune system, and in particular you can design different models. You can add the physiology, you can add the disease, and you can add the treatment, and you're also allowed to investigate the effect on vulnerable population that, for example, with the in vitro method you are not possible to study. And we decide to consider different age of exposure. And today I showed you the results for the young people. And to do that, so we use the PBK model results, and we insert in the Universal Immune System Simulator, and we simulated two different scenarios. The first one is a general bacterial challenge to see how the presence of PFAS can have an impact when we have a alteration of the immune system. And we also mimic the effect on a particular vaccination. Before I show you the results related to a general bacterial challenge, and you can see here that we obtain a statistically significant difference, and in particular suppression, all of the four more important parameters for the immune activation, only in the exposure scenario 3. So the one that in the PBK model was represented by the blue line. So only in the one we are in the people that live in contaminated area. And we see also in line with the results that we obtain in vitro, a reduction of interleukin 17, but also a reduction of the of EGM expression. So that result give us at the end that the threshold that EFSA pose based on human epidemiological data also work in a good way in our model. And we do the same also mimic two different typology of vaccination. The first one is a general influenza vaccination in which we have only one injection. And again, you can see that all the scenario 3 was able to suppress in a statistically significant way the efficacy of the vaccination. And in the same, we have also the reduction for a second vaccination that is anti-diphtheria 1 that have different compared the influenza 1 because it's 2 injections, but also in that case, in the second injection, we obtain a statistically significant reduction compared to the control for the scenario 3. And when we cover the kinetics, the dynamic, and try to go on and predict more the in vivo human relevance, also adding some, for example, in immune dysfunction or vaccination, we decide also to use again the PBK model to try to perform the quantitative in vitro to in vivo extrapolation. So again, starting from the PBK model, we decide to apply not only the nominal concentration using the in vitro system, but also to take in consideration that our PFAS can link protein, lipids, plastic, and so on. So we ran a in vitro distribution model, and we saw that in our model, experimental model, the PFAS link in particular the protein, and so we decide to correct the nominal concentration used in the in vitro system with with the protein binding. So we here below you can see the formulation. In the in vitro system, I didn't use the FPS, but I use a human serum also to reduce the animal model present in our protocol, but also to increase human relevance. So here you see the concentration that we use, and we correct the nominal concentration for protein binding, since our PFAS basically link quite all the albumin in our in vitro system. Then we run the concentration in the PBK model to convert the in vitro concentration to an oral equivalent dose, and then we perform the benchmark dose to obtain an oral equivalent effect dose. And the results that we obtain for the single PFAS are here represented for both male and female. But we decide to make a sum of them because in the EFSA Contampane of 2020, the tolerable daily intake was based on the 4 long-chain PFAS altogether. So if we made the summary, you can see that with our in vitro system and using the PBK model, we reach at the end the same, more or less the same range of oral equivalent dose. So we can assume that at the end we was able to mimic the in vivo condition, and also to reach with only in vitro method the human epidemiological data obtained for set up the tolerable daily intake by EFSA. So we decided to use that strategy, so to the effect on T-independent B cells activations on the one just very brief with the ODN 2006 stimulation to obtain the results in the in vitro with the in vitro method, and to then move develop a PBK model to then have a confirmation in universal immune system simulator and obtain the benchmark dose for the quantitative in vitro to in vivo extrapolation. And starting from that strategy, we want to go on with other PFAS. So the conclusion of our work, and that basically I have already discussed, is that the data obtained support the in vivo evidence that are right now present for the PFAS, so immunosuppression, and in particular the NAM that we decided to use was able to mimic the in vivo condition and also evaluate the effect of PFAS. And the main effect observed was for sure a clear antibody production suppression. And we also found that we are able to see a different regulation of the marker of the cytokine involved with different time and also modulation. And concerning the Silico method, we was able to add some information that with all in vitro method was not possible, so add information related of the dynamics and the kinetics, and also to at the end reach the same oral equivalent effect dose that was based on epidemiological data. Moreover, for sure also the use of the universal immune system simulator was very useful to then try to mimic and reproduce the human real life. And overall, our project aimed to develop a property in vitro in silico method, also to not cover the data gap that was identified by EFSA, but also to maybe in future screen more than 4 PFAS using the best testing strategy that I showed you in the slide before. But what are the future directions? So now we are move on to study also the effect on short chain PFAS using the strategy that I showed you before to try to screen a big number of PFAS, and which are the different for now. So seems to be more soluble, and also to stay less in human. So starting from that evidence, we decided to perform the in vitro distribution model, and we saw that effectively they link less the protein and stay more soluble in the water, so in our medium. And we saw also a reduction as likely effect, a less suppression effect compared to the long one. I cannot show you because it's part of different project that is not published right now, but I can anticipate it that. And so maybe the less effect could be also related to that, because they enter less into the cells and stay more into the medium. Or also we want to try to develop all the PBK model, to study the PBK model also for that short chain PFAS. And for that topic is super important to study the reabsorption, that for the long chain, as you can see here, it's very high because there is a higher half-life, so the PFAS, the long chain PFAS returning continuously into our body and stay, while the short chain was immediately eliminated due to the solubility. So that one is our also next point, so to develop a properly PBK model also for the short chain PFAS. But that one is at the end is another story. So I want to thank you all of you for the attention. I hope to give you all the information. And in particular, I want to thank all my laboratory in Milan and Professor Corsini and all of the people that work in the EFSA project, because I'm more in vitro girl and I have the opportunity to work with SQ Lab, in particular Licia Paini, and also Professor Francesco Pappalardo for the in silico part during my PhD. And so I'm very lucky to was able to cover both of the world, both in silico and in vitro. And I finished. Thank you.
Okay. Thank you very much. That's really impressive to put the simulations and the experiment together that way, and to chain the simulation work so that you get really the full relevance. Time for questions. Chang, do you have a question?
Yeah, I have a question. Hi, Martina. Thank you for a very interesting talk. Putting all these PPK modeling, in vitro assays, and also the dynamic model together. So my first question is, about your assays. I'm not really familiar with a particular T-cell dependent versus T-cell independent assays you're deriving here. But normally when people talk about T-cell independent B-cell antibody production, we're talking about mostly the IgM type of response, right? And for the T-cell dependent, we're talking about more like a non-IgM or IgG type. But in your assays, I see for your TD response, you're actually measuring some anti-IgM for your T-cell dependent. Well, for your T-cell independent, you're actually measuring IgG and IgM. Can you clarify on that?
Yeah, so for the T cells dependent antibody response, we prefer to measure IgM since that one are the more present, and also for very simple ways. So we found all the analyses that detect anti-KLH IgM. So now I saw that a brand developed also one for the IgG. I bought that just to test, but it's not so in the T dependent are more present IgM. And while for the other one, we decide to study both just to see if they have a different impact.
Okay, so basically in your case, to differentiate the two assays, other than what's measuring, it's the stimuli, which is different, right? I noticed for your T-cell independent, you use the CpG, right? And then for the T-cell dependent, you are using the L2 and or some interleukin.
For the T cells dependent, I use as a antigen the KO lymphetemochanin, so KLH, while as a sort of stimulus to increase the proliferation of B cells, I use the Staphylococcus CoA1. Staphylococcus aureus CoA1, that since it's not toll-like receptor 9 agonist, is able to help me to increase more the number of B cells without any production of the antibody. While for ODN 2006, we have the production also of the antibody.
Okay, thank you on that. So my next question, I'm assuming other folks are going to ask you the same question. What's in that universal immune system simulators in there, essentially? For that, basically, simulators, what's actually our essential process is actually being modeled. Mm-hmm.
Yeah. Actually, I didn't use it because it's from the University of Catania. I just sent the results to Professor Pappalardo from the PBK model, and they ran all the simulation and give me back the results. So for that one, I just know the base of the model and the system, but I never see also the platform and so on. So
maybe we can have you connect us with somebody who could present that as a partner talk to this one. So you've shown mostly the experimental work, and then we could have somebody else show us a little bit of the detail of the simulation approach.
Yeah, sure, because they use for me and for the project in general the universal immune system simulator mode with the model tox that was developed to study endocrine disruptor and in general environmental contaminants, but they work a lot also to study sensitizers, so they have also a tool for study sensitization, so I can give you the name and also connect it for sure.
Other questions? I have a basic question. When you see that effectively I assume you screen for this, when you see that all of the PFAS cause this the loss of these immune responses, how do you check that it isn't something simple like cytotoxicity? Because you're using, as you said, you're using fairly high levels of exposure.
Yeah, I check all of the time the cells, the cell viability, and there is no cell viability. So in reality, I know person from literature that work with a more higher concentration than me. Some people work with 200, 300 microgram per ml, so I stay only on 10. But in any case, since I have a very long exposure, because I reach one time also 11 days, I check it, but I have no cell toxicity. So, and now that I change PFAS, so I use another typology of PFAS and I see a different results, I can trust again more in that because I see different effect.
So you mentioned that you used source cells from male and female populations. Do you see, could you do, at least in the future, more fine-grained work to look at people who might be immunosuppressed for some reason, or different
Yes,
ages of the individual from whom you had the samples to
yeah, it's something
investigate?
that yeah, for sure it will be interesting. Unfortunately, in Italy, it's not so easy to take that information. So, for example, I didn't know the exact age of my donor. I know only that are healthy donor with the age between 18 and 75 years old. So, for endocrine disruptor in particular, chemical, sorry, but so in particular PFAS, and also for hormonal behaviors, hormonal status, for sure are information that in the future, if someone have the possibility to have it, will be fantastic. Also to understand the immune function of the people. What I can tell you is that all of the time I didn't mention, sorry, but you see in the slide, if you want to check again in the registration, I use a positive control that was a no suppressive drug, so cyclosporin A or rapamycin, or for example dendritic cells, dexamethasone, just to be sure that the immune system of the people, the donor that I use, are work and didn't have problems. So I use only at the end for the analysis people that have a properly effect on the positive control. So just to be sure to work with people that don't have a problem with the immune system. But that one will be for sure a very interesting information to add it.
But it might be, might be interesting that people with other immune suppressive conditions might be more or less susceptible to this kind of
Sure.
immunosuppression you're seeing here. Presumably, I don't know, are there data sets, sort of classic RNA-seq data sets, showing how to try to get into more of the mechanistic detail of which pathways within the cell are being affected by the PFAS exposure during differentiation?
Yeah.
So you could do differential RNA-seq between the PFAS exposed and the non-PFAS exposed cells in your samples.
Yeah, we have also that results actually. So you can find all in the report on EFSA journal. So the people from Wageningen identify two major pathway. So the PPAR alpha pathway and the glucocorticoid pathway for both dendritic cells, T cells, B cells, also depending on the timing. And right now we are running some experiment to confirm that involvement also using real-time PCR from the most important alterated genes, and also I running some experiment ad hoc to block the way. So, for example, use an antagonist of the glucocorticoid receptor to block the way and see if in that case I didn't have the effect or I have it, so just to exclude or not the pathway. But we are working also on it.
If you have differential gene expression, one of the people who's usually here but is on a plane today, Rita de Almeida, has some nice software for doing the analysis of those differential gene expression that might pull out a little bit more information about differential pathways than using standard methods. So we could connect you with her if you're interested.
Okay, yeah, thank you. I'm not more familiar with RNA-seq because it was performed from other people,
Right.
but for sure I can have a look.
Lorenzo.
Hi, thank you very much for your talk. I really like this approach of blending NAMs in vitro and in silico to try to address things that so far have been addressed in vivo only. Well, my question is about perhaps a little technical, but we know that whatever substance that goes in the blood, goes somewhere, somehow goes in the blood, is absorbed, etc., may or may not have a certain binding with plasma proteins, and that might modify the activity. Sometimes it increases, sometimes it decreases. So have you considered that, especially I really work a lot in vitro, have you considered, tried or thought about using serum with or without reduced serum condition where you might be able to address this
Yeah.
aspect?
Yeah, so from technical point of view, first of all, I use medium that is without phenol red, since PFAS are endocrine disruptors, so phenol red can maybe interfere with the hormonal receptor. And then I use for the for TH1 cells, I use the fetal bovine serum delipizide. So ad hoc for don't have so much protein and lipids inside. And as I told you before, for the PBNC, just to move in a alternative completing method, I use a human serum. That one is not possible to have it the lipizide, but I run the in vitro distribution model. So I also study and try to see how my PFAS link, and for the long chain PFAS, the link basically for the 80% the albumin, 2-3%, then depend of the PFAS lipids in our experimental condition, and the 10-20% enter into the cells. While for the short chain PFAS, it's completely contrary. So the most ones stay on the medium, also on water, not linked to nothing, and a few enter into the cells. And only PFOA is a very, very small volatile between all. So we also add that information just to see how they move in my in vitro system. But yeah, we try to take in consideration and also reduce eventually protein, in particular albumin binding.
And there is some evidence, I don't know, from toxicological studies where, so these data, I assume they are kind of mapped to the in vivo situation. And is there any other evidence that these are, I don't know, there in the literature of binding curves in vivo where you can say, oh, these are the best condition for me in vitro, and that's rely on.
For the condition in vitro, we already use the lipizide serum for endocrine disruptor in a old project, and we use also a less concentrated, so we use the 5% instead of the 10. Concerning the in vitro distribution model, yeah, I know that other research work on it, especially on PFAS. Just one name, Proenka et al., an article from Proenka et al., study in a different, not in immune system, but are present in literature. Also, Steliaki, Pratki have something related to that. So yeah, at the end we are in line with also the in vitro distribution model that they perform. And concerning the use of serum, for now I didn't see so much in vitro results that use the other serums. So also go around in the different congress. Also, for example, for the medium, the people was very surprised that they used the white one instead of the one with phenol red. So maybe we are move on also in that way in general.
Good. Thank you very much. It was a great talk. Thank you.
Thank you.
James, I have a question. This is Tom.
Please go ahead, Tom.
Sorry, I missed the beginning of your talk, but so I just retired from the EPA, and of course, we've been working on NAM-based approaches for lots of different things. I'm not that familiar with the perfluorinateds, but I do know that there's a highly sexually dimorphic toxicokinetic difference, at least in rats. And I was kind of surprised that you're, at least if I interpret it right, you didn't see much difference between the male and female responses. Have you looked for those sexually dimorphic differences in your toxicokinetic profiles?
No. So I know that all the data insert in the PBK model that I didn't write, I just ran it, are based on human for P4 and PFAS and monkey for PFNA and PFHXS. And I found for all the literature results in rat more male impact compared female. So here we found a completely different, so male female, but I have no other idea or.
Yeah. Well, certainly if you did the PBPK using an animal PBPK model, it would be interesting to see if you get validation against what's known for the animal studies. Yeah, the other.
Sure, but maybe I can ask to the people that are able to design the PBK.
The other question I had on a similar note that James was talking about, but have you considered the impact of PFAS exposure on early prenatal development? So I know developmental immunotox is a big concern with the perfluorinateds, and I wonder if you looked, say, in a pregnancy PBPK model, or if you know of any literature looking at hematopoietic stem cell differentiation that's been exposed to these perfluorinated compounds. Do you expect to see effects on differentiation?
I don't know, maybe. I don't know. I have no idea.
Okay.
Maybe yes, maybe there is an impact also.
Any other questions for today? I think we had a good discussion, and
Thank you.
I really appreciate you coming, and especially for this slightly awkward time of year.
No
And
worries.
it's a very exciting piece of work, and I hope as it develops and you move on after your degree, perhaps you'll come back and give us an update on your research. Because this issue of NAMs and connecting NAMs to systemic physiological outcomes is so important, and it's so difficult to do those chains of inference, to do in vitro to in vivo extrapolation. It's really exciting to me to see this work. So I want to thank everybody. I want to thank everybody for today, and I'll look forward to seeing people moving forward in this year.